We started this in our little collaboration in 1979, which was what, I guess about 3 or 4 years after I first started in the LTP business.
Well, your first paper, was 77.
77.
Yeah.
77. So yeah, you just came.
Yeah, I just came just at the very early days.
Right.
Yeah.
See, those were days of one of my many colossal miscalculations, not counting taking you on as one of them. That wasn't colossal. That was just one of a many long series of. But the colossal miscalculation was, and it's a great illustration of scientific history, was that we were in a race. Now, I was convinced we were in a race to master LTP, that the whole planet would be, hordes of scientists would be pouring into LTP. So the lab was, as you were coming into it, and I'm sure you remember, was just a beehive of activity. I mean, it was mobs of people because I was cracking the whip and saying, man, we are up against the best and brightest in the world. This is no time for pause, no time for...
No time.
No time. 12 hours a day going on. That was just a monstrous miscalculation. There was actually no market for LTP at all. The number of people that were really charged into it was tiny. And, it was just a giant miscalculation. And Richard Morris is the English neuroscientist has often said that, he gives us credit for being as much as anybody, the people that actually made LTP a tractable subject for people to get into. To have fun with.
That's true.
But Tim Bliss didn't follow the inventors of it, dropped it. It was just incredible. So Michel comes in, and at the very beginning of it, it was obvious, and it was why it was such a delightful scientific topic, was that it was constrained by if you thought that it was the substrate of memory, then it was constrained by the properties of memory. It had to be a certain way. There had to be this kind of answer, and that made it a really nice problem. And then you came in with what seemed to be, and what we both went on to convince ourselves, was the big part of the answer, namely that the effect was going to be an increase in glutamate receptors, right? Yes. I don't know how you got started on glutamate receptors.
Well, I got started because when I was doing my Ph.D., I was working on dopamine receptors. And we had worked on a kind of analogous story to the LTP story in the field of dopamine receptors. Remember we had this long-term facilitation of dopaminergic transmission. And there, I was convinced it was due to a change in dopamine receptors. So when I started reading, the LTP story, I immediately was convinced that this was a change in the receptors at these synapses, which was the glutamate receptors.
Right.
And indeed, immediately I jumped on the glutamate receptors, and that was also a second mistake. The second mistake that we were measuring this glutamate binding, and we are convinced we are looking at glutamate binding to the receptors, and elaborated the whole Calpain story and everything, and it turned out that this glutamate binding was not to the glutamate receptors.
Right.
So that was
giant setback.
Giant setback.
But the odd thing about it is, and of course this is the way the history of there's the fascinating thing about the history of science to me, and it doesn't matter what field you look at. Okay, there's a great book out there by a guy named Ohanian called Einstein's Mistakes, and it should be mandatory reading for everyone interested in the history of science, because if you look at the Einstein story, it's all you see are the triumphs. You never see all the stuff that went in between. And that's what he's written this book about. Okay, so it shows that, no, this was dead wrong. And oh, no, that was way wrong. And no, there was a huge emotional battle with these people and all that. That's all lost to history. To history, all there is what worked out right.
That's right, yeah.
Okay, and that's very much the way it is with the LTP story.
Right.
I mean, if you look, certainly 95% of the stuff that's been published on LTP over the years is just the giant dirigible came and picked it up one night and dumped it out in the Pacific Trench.
Right.
I mean, we only remember, and that's the funny thing about it. We were so, as it happens, we were completely right about everything except what we were measuring.
Exactly.
In other words, it is glutamate receptors.
Indeed.
It is Calpain.
Yeah.
It is an actin cytoskeletal change. All that stuff that wound up in the 84
science
science paper,
Yeah.
I still use that figure introduced my talks today to say, yeah, this is what LTP is about. And it is all there, down to the drawings.
Yeah, that's true. I think, I reread this science paper, and we were so ahead of our time. And it is unbelievable that today you can read it, and it sounds so true and so real.
It's funny, it really is. And that was, again, because I think in some measure both of us were genuinely interested in this thing as a substrate of memory, and we were asking the question, what would be available? Memory is one of the most peculiar biological traits you're ever going to come across. Just saying that something can happen in a flash and last indefinitely and has infinite capacity to it, how many mechanisms, Johnny, are you going to find that are going to satisfy those constraints biologically, and has to be driven by a naturalistic rhythm? So you remember shortly after we did the science paper, up came the theta rhythm thing. We linked the whole thing together. So we had a naturalistic rhythm, all the mechanisms we worked out. But we were constrained by those memory problems. And I think those things led us to what it is.
Right.
And what other people did was they chased it as it existed in their own minds, rather than looking at it as something that must fit these constraints.
Right.
And I think that really did guide us to the right answers.
Yeah, well, I think it's also because we were in some ways naive.
Yes.
But we were not really the molecular biologists, or we were not really, this or that, and we were not bound by any of the past. I had nothing to do with, with it before.
No.
And you had such a great open mind, and that it was indeed, this kind of free-floating atmosphere. And I think it was also the time,
it was.
And the Irvine ambiance at the time, that it was really kind of more of a kind of scholar place, that people were there really interested in the science.
Yeah.
And independently of a sociology of science.
No, that's dead on, and that's a very good point. Those were years in which the idea component of science, the romance of science, was larger than the sociology of science.
That's true, yeah.
Not just at our place, but other places.
That's true, yeah.
But it was very intense at Irvine in that regard in those days. I mean, that was
But it's true, the Irvine, I think to go back to, to this Irvine attitude, I mean, it was indeed both, the competition between, the Cutman and the Lynch and the Mango, but at the same time it was this collectivity of really, intense, dedicated scientists. I always remember, the day that the campus was flooded and the cops came and said, everybody goes home. And everybody in the lab said, we are not going home. We stay here, we keep working, we'll sleep here if we need to, but we are not going home.
Yeah, I remember this. No, it was astonishing. And that was part of the era, I think. But it was very intense at UC Irvine. I don't really know why. Mostly it was, I suppose, a lot of it was the major figures were young scientists. And it was also something about a new campus. But the energy and romance about science, about the results, about living in the results, and versus the sociology of it was so different then than now. I mean, there's so much more sociology. I think it's inevitable when things get large. It's hard to imagine now what the size of the Society of Neuroscience was in those days. I mean, what, 10% of what it is now? You knew everyone. Yeah. You would go to a European meeting and you'd get off and you knew every single, so.
But even, the first LTP meetings, I mean, there were, 50 people.
Yeah, max.
And everybody knew each other, but although it's true, it got a little bit derailed, with this argument, the pre versus the post-synaptic argument. But at the same time, there was still, it was contained within, a small community, and people were still, indeed, still focusing on still trying to find the answer.
Yes. There's a real question as to whether or not any organization, when it grows very large, can maintain that kind of, this is a question, of course, that comes out of industry all the time. Can you take a company that is a dynamic, small company and maintain that kind of dynamism when it gets large? It's the same question we should ask about neuroscience. When you take a thing like we had in that time and place where, And also the relaxation of it. Put it this way, remember the number of students that we would have come into that lab as graduate students that would be accepted into graduate school for Ph.D. programs. Those guys wouldn't have a cellophane snowball's chance today of ever getting into a major graduate program. I remember Mike Browning. I mean, we had a collection of graduate students that were just, I mean, they were like out of a novel.
That's true.
I mean, this guy Browning came in, and his main claim to fame was he was an English major who had listened to a lecture I gave at UCLA and decided that was his life work and just basically moved in. And one day we found out, right, he was doing experiments, and I mean, it was amazing, and we got him in graduate school. That sort of thing is impossible today. It's today the department would, and in fact they stopped accepting those kinds of crazy graduate students of mine.
Yeah. No, but I think also the collection of undergraduates. I mean, you had also fantastic undergraduate students, and again, I think there was this whole notion of the excitement of science, the excitement of research, the excitement of participating in something big. And I think it was, everybody knew that this was a big question. I mean, there was no doubt. And you made it, you made everybody indeed strongly believe that this was indeed a big question and that, there would be big reward to solve it. And I think that message went through. And really, I think it's still, to this day, to me, I think it's still, a great motivator.
Oh yeah, it's harder today. It's much harder today to maintain it because what goes into a publication today is so much more elaborate. There's so much more collaboration that's necessary to do it. It's an open question to me as to whether or not it was a mistake to move away from the kinds of publications that people did where they basically were result-driven publication. What you do is you have a result, you publish the result. Today, papers are much more elaborate. It means so many more people have to be involved. There's so many different groups. It dilutes the enthusiasm that the single student working on getting a single result can have. I mean, back then as now, the student might go to this lab to get this piece of it and that piece of it and go across all this collaboration. But in the end, they publish their result. Today, you publish, I don't know what you would call it. They're much more elaborate papers. And in some senses, that may be one of the factors that changed. But yeah, Irvine was fascinating for the quality of the undergraduates. I mean, for years, we were supplying some of the best graduate programs in the world with our undergraduates. It was amazing. I don't know what that was about.
Yeah, but I think it was the whole community. I mean, I think the whole psychobio. I mean, I think the whole psychobio at Irvine was really, probably one of the, best place for neuroscience.
It was.
And, I don't know what happened to.
Well, there was a campus that had. It was a campus that had 5,000 undergraduates, 2,000 of whom were in biology. And it grew to a very large campus. And again, this is the eternal question of size.
Right.
There's a great satisfaction in the whole thing. I mean, to all the people that were there at that time, they all remember those episodes. I mean, every time you go to a neuroscience society meeting, you run into somebody from that time, and they remember one more insane story, one more all-night experiment, one more. If we can just get this to work, then we'll have, all those things. And there's an incredible satisfaction that goes into science. And I think you and I, to some sense, exemplify this, illustrate it beautifully, I think, that those things that you did then are still with you today.
Yeah.
There is a satisfaction to that really cannot be described. It wasn't that you worked on this project, then you moved over to this thing, then you moved over here, and then you did this and you did this. The feeling that your life is continuous across that number of years is immensely satisfying. And maybe it's the biggest satisfaction of it all. I mean, we are two happy campers because, as it turns out, we were pretty much right 25 years ago. It is a change in the actin cytoskeleton leading to more glutamate receptors in the postsynaptic spine. And at the time, that was a radical, crazy thing, and most of those words weren't even known to most neuroscientists.
That's true.
I mean, and it turns out, that is LTP, and that's the way it works. But it's to me, is critical to it is the business that it is the same thing. It is the same. The continuity of it is so important.
Yeah, no, I agree. And in fact, it's interesting because I'm telling, all the students who come to my lab now, I said, this is a great time because now we are closing the books.
Yeah.
We are finishing up the story that was started indeed 25, 30 years ago, and we are closing it. And within the next 2 years, it's going to be finished, over, understood, everything.
I agree. It's absolutely astonishing to be saying those words. But we have, in the last 3 years now, reached the point where we can actually visualize the synapse changes that we put into that drawing. I mean, the day 3, 4 years ago, whenever it was now, that I actually saw those increases in synapses and all that stuff we had in that paper, it was just, I mean, there is no word to describe that. I mean, there it was that many years later, and we ran a study, and now with all these new microscopes and technology, boom, there is the change that we had predicted.
Right. Yeah.
And moments from now, we're going to have, there will be no more receptors.
Yeah. And as I said, the day, we saw, calpain activation in the spine, I mean, it was also such a great moment because you said, finally, after 25 years, to be able to see it.
Yeah.
That was indeed, such a reward.
I don't think anybody has a clue either as to how much of that stuff we pulled out of the air. I mean, my God, calpain. I mean, how many people had ever heard the name? I remember going to a meeting and people thought that was something that we had come up with using the word California. Calpain, we get it. That's one of your silly, stupid adolescent jokes. It's the pain you're inflicting on it from California. No, we didn't come up with that word. Baudry and I, because we were the only people on the planet talking about calpain and synapses.
We had a couple of Japanese.
Yeah, that was it. Well, the guys didn't found the damn enzyme.
Yeah.
But that enzyme was selected because there had to be an enzyme respond to calcium. We could reorganize the spine cytoskeleton.
That's right.
And so there was only one candidate, and there it was, and we said that's it, and went off, chasing it. But that it turns out to be right. Yeah. It's just astounding. And it's not so shocking about the glutamate receptors because what else was it going to be at the end of the day?
Yeah, exactly. Yeah.
Really, when you think about it.
Exactly. Yeah.
The whole presynaptic thing. Sure, but. It had to be one of those two, and the odds were always heavily on it being the glutamate receptors. Pache, Eric, and all the rest of the guys. that was the far and away the likeliest candidate. But to think that we picked calpain of the thousands of enzymes that are sitting in that spine and say, it's amazing to me. Yeah. That will always be. We talked about that quite often. Also, the fact that even to these days, people are still questioning, the role of calpain in LTP. To me, it's still, and that very few people, still put calpain in their models of, LTP. Still, to me, is astonishing. Well, it's because again. And that's when you look at the history of science, you just don't realize the extent to which things become big because a context has been built for them. Mm-hmm. Yeah. And the current context of this field is not one of elaborate, despite what people like to draw, is not one of elaborate pathways. It almost has to be linked to something that is currently right now. Yeah. And you've tried to do that, but it's the context that's the issue. It's an amazing thing, but people, you can't imagine it. You can be dead on. You can have it exactly right. And unless there's a market, where a market means it fits into the current model, or the current, not even the model, the current talking. The current talking, whatever the current talking is. Now calpain is going to have its day because what's all about LTP now is the cytoskeleton, and calpain is the owner and operator of the cytoskeleton, so it will now have its moment. Some God knows how many years late now. It is very hard to remember this thing about looking back on the history of science, and I've had a number of people talk to me about the history of LTP. All they're looking at is the mountains over the fog. They're just looking at the peaks of the mountains, and they're saying that's the history. The history is down in those valleys. Down in those valleys are where the bones are. That's where all the losers are. That's where all the terrible ideas are. And those valleys are half filled with debris of the mighty army of LTP as it went through. And all they're looking at is those peaks of the mountains. They don't see anything underneath the clouds. It's like that for all science. It really is. I mean, that's the way it works.
That's true, yeah.
So true. You got to kind of like hope, that your mountain is there while you're still alive.
That's right.
They're still around.
Right, yeah.